Now, basically, we were talking, we were saying that we are done almost with the crosses, but the thing is that we need to practice them with different angles. So overall, let's have a very quick revision. That we said that we have different terminologies like genotypes, like phenotypes, and we said that meiosis separates the alleles. The alleles recombine. This is the genotype. This is the genotype, and their appearance is the phenotype. And genotype is made of two alleles that we separately get from our mother and father. One allele we get from our mother, and one allele we get from our father. And again, when we try to make the gametes, or when the gametes were formed in our parents, and the gametes are forming inside us, the alleles have to separate, because it is a rule of meiosis.
Your voice is cutting quite a bit.
Random type of you can say random fertilization is the true word that we should be using here. Okay, so next is that once we are done with it, we did the crosses in two patterns. One, we did it from parents to offspring. We said that when the parents are there, their gametes would be separating and then they would be combining. The second case that we built was, we said that a couple has three children, two with attached earlobes and one with detached earlobes. Draw the genetic diagrams to show the genotypes of the parents. We said that the attached earlobe is a dominant factor, is a dominant phenotype containing the allele uppercase D, and the recessive factor is is expressed by the detached earlobe, and its letter would be a lowercase d. Now, the thing is that when we try to make a cross, when we try to know the pattern or the what I must say, yeah, when we try to know the pattern or the genotype of the parents, we need to move in reverse. How? We prefer a person who is showing the recessive phenotype. Obviously, if a person is showing the recessive phenotype, it means that that person would be having both recessive alleles or both lower cases. And we know for a fact that if a person is containing two recessive alleles, this person is getting one recessive allele from one parent and other recessive allele from other parent, meaning that it is obvious that both the parents contain at least one recessive allele. At least one recessive allele is being possessed by the parents. Second, their children also are showing the attached phenotype, meaning that one or both of the parents are also they may also have a dominant allele. Why? Because if two of their children are getting a dominant phenotype, it means that they must be getting a dominant allele from their parent, and it means the parents possess the dominant allele. So we said that maybe both of the parents are getting the are having the dominant allele, and which is going to appear in the offsprings. That is basically the way we do the crosses. Is this clear?
Yes.
Okay, moving on. This type of cross basically is called as a monohybrid. What is a monohybrid? Remember, now we need to clear out some terminologies which are left, then we'll be moving forward. A monohybrid is a cross. It basically is a terminology that was generated by the father of the genetics, called as whose name was the Gregor John Mendel. The monohybrid cross is a cross in which we only talk about one character. We are not talking about two characters simultaneously. Not talking about about more than one simultaneously. What is the meaning of it? Suppose there is a character we just discussed, uh, ear attachment. Let's say we start discussing another character with it, which is ear structure or the earlobe style. One character is attachment or detachment. That is one character. And on the other hand, the other character is the uh, the size of the earlobe. Now these are two different characters. If we study them both simultaneously, that would no more be a monohybrid. That would be a dihybrid. If we start studying three characters simultaneously, that would be a trihybrid. So monohybrid crosses are all those which we often talk about when we are going to study only one character per cross. One character per cross. Is it clear?
Yes.
Moving on, there is now one type of cross that we discussed, uh, a character. Okay, hang on. Types of monohybrids. One is simple dominance. What is the meaning of simple dominance? Simple dominance means that a gene containing only two alternate forms. What do we call the alternate forms? Alleles. A gene containing only two alternate forms, one dominant over the other, just like A is dominant over A. That is called as simple dominance. Okay, there is another feature, there is another type of dominance in monohybrid crosses. But before that, I would like you to tell me, how did we say that the alleles are formed? Alleles of a gene are formed.
Sorry, could you
Like there
repeat
is
the
a,
question? How many alleles are
suppose there
formed?
is a, how alleles are formed.
Um, so
Suppose,
mutation.
let me give you an idea. Yeah, very good, very good, very good. That's fine. That's the complete answer. Okay, so the alleles are formed due to mutation, meaning that if this was the original gene, now it has this alternate form because of the mutation. The question is, does mutation only give rise to one alternate form? Only give rise to one alternate form? No, the answer is no. It can give rise to more than one alternate form. More than one alternate form. And as a next step, I would say that it can give rise to more than one recessive or dominant alleles. More than one recessive or dominant alleles. Now, what is the meaning of it and how does it relate to the previous concept? Suppose this is a gene M. Suddenly what happened that this gene gets mutated and get a slightly alternative, I'm sorry, a slightly variable protein, because we said how the genes express their difference. The genes express themselves with the help of protein, right?
Yeah.
So obviously, if the genes are expressing themselves with the help of proteins, it means that the genes are the genes are also going to the genes are also going to like uh, express their variations. The genes are also going to express their variations by by showing different proteins. Obviously, they are going to express their difference by showing different proteins. So the M gene mutated, the M gene mutated, and this M gene is um, this M gene is showing slightly different protein. It is showing slightly different protein. So it means it is an alternate form. Then this M plus suddenly gets mutated into M star. And then this M gene gets mutated into small m, lowercase m. Now, if you notice, you have three dominant alleles. Three dominant alleles for same locus. Like either of them can occupy that locus. Either of them. Let me open the whiteboard. Either of them can occupy the locus for M gene. Meaning that you got the link?
Yeah. I'm saving it.
Okay, I'm changing the privacy settings. Okay, just check you can access it or not. So basically, we are now having more than one allele because of constant mutations, and they are going to occupy the same locus. Remember, no matter how many alleles, no matter how many alleles are there, no matter how many alleles are there, they are supposed to be the same locus, occupying the same locus, and occupying the same locus, and uh, there can only be two alleles per person. Like you may have 100 alternative alleles for a gene. Let's say A plus, A star, A. But a person can only get this combination A plus maybe, A star, A plus maybe, A star, lowercase a maybe, or A. Like multiple allele does not mean that they are going to accumulate in one person, but it is a reality that this phenomena is called as multiple alleles when you have more than two alleles for a character. When you have more than two alleles for a character, that thing is called as multiple alleles. You got it?
Yeah.
Okay. Now, if you notice, the A and A plus both appear to be dominant, don't you think?
Yes. They are dominant.
So how would they express? How would they express? Remember, none of them would hide the effect of others. None of them can hide others' effect. Others' effect. Both express themselves both express themselves clearly. Clearly. This phenomena is called as codominance. It's no more a simple dominance. If you go back to the previous Jamboard, you will see simple dominance means that A is dominant over A. This is simple dominance. Right now, both are dominant. So you would call them co-dominating alleles because A plus, both are completely expressing. A star, A plus, both are completely expressing. A star, A, both are completely expressing. That is why this phenomena is called as codominance. Now, leading you to the example, blood group. There was a time in the very far past, there was a time the blood group gene was only one, and that was I. I basically was the alphabet given to the blood group because the blood group basically makes a protein called as isoglutenin. This is out of your syllabus league, so you can just forget it. I is the gene that we select for the blood group. Now, we know that how many blood groups do we have often in a population?
You mean like the OA type?
Yes, 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 yes. I'm not talking about plus minus. Suppose
Four,
if you
right?
have,
A,
yeah, if you had a
B,
gene
and
A,
O.
B, A, B,
Mm.
O, B, A, B, and O. If you had a dominant allele for blood group, and let's say this allele is expressing A blood group, and then there is a recessive allele, and it is there to represent O blood group. What does it mean? It simply means you wouldn't have more than two blood groups, right?
Yeah,
So how is it possible that there are more than two blood groups? It could only be possible in case of multiple alleles. See, the I gene is now present in the populations in three different forms. IA, that would be coding for A blood group, IB, which is another dominant allele coding for B blood group, and IO. Basically, you were supposed to be writing it small I, but your syllabus says that you should be able to write it in IO form. IO is recessive. I would have preferred that I should be writing the recessive in the lowercase letters, but the syllabus says that the IO. So let's say IO. Then IO is showing the O blood group. Now, let's suppose I say that if I ask you to make the genotype, I am saying this is the column to mention alleles. This is the column to mention the groups. Now, the third column, let's make the genotypes of these blood groups. What do you suppose would be the genotypes for A blood group?
so haven't we done A already? So there's A on top. Oh, okay, wait. So
Now
genotype
there are
for,
multiple genotype. You remember the genotype?
so AA,
Okay, IA. You have to present it like this because these are no more single alleles. These are multiple alleles now. Any more genotype you expect?
A, so I, A and IO.
Well done. IAIO. Second, the same. IAIB, and IBIO. These are the expected genotypes for B blood group. For O, what do you think? What should be the genotype?
For IO, hmm, so IO, IO.
Only, yes. Very good. The fourth blood group is AB. Now, what genotype is going to be presenting it? Both dominant allele combined per body. Both dominant allele combining in one body would be coding for the AB blood group. So this phenomenon now, see over here, you can clearly see. Look at the circle genotypes. These are clearly showing simple dominance. This is simple dominance. And IO is showing no dominance recessive relationship. Why? Because both are same. But these are showing codominance relations. Is that clear?
Yes.
Now, the codominance basically have multiple examples. No doubt, there are many examples that are covering the topic of the codominance. And we have a worksheet. That is why I was immediately asking you, because I have more worksheets for this chapter. I have almost 4 different worksheets, or maybe 5. Yes, 5 different worksheets for this chapter only, because this is one of the toughest chapter. Okay, so now, moving on, they've been talking about, like, obviously we are done with one more terminology is still there. I need to cover. We have covered codominance. We have seen the ABO blood group system. Sex-linked characteristics are there, and then red-green color blindness. Okay, genetic diagram to predict the results of monohybrid crosses. Obviously, I have shown you how to calculate the phenotypes and sex linkage ratios. Yes, that is the so this is not done. One portion is not done. These are done. Okay, this is to be done, and we'll be doing it, covering it inshallah today. And then one more thing, uh, test cross. Yes, test cross is remaining. 2 terminologies are still there, 2 to 3. There is a terminology that you would find in your syllabus: true breed. What's a true breed? A generation of plants, mostly, that is constantly showing constantly showing homozygous features. It could be homozygous dominant or homozygous recessive, homozygous genotype for several generations. Like, in their generation, in their whole family, there is no plant that is representing the heterozygous trend. But the true breed only is the terminology which would be covering only those organisms, or only those plants, or only those animals which are showing the same type of characteristic for a long time. It could be homozygous dominant, or it could be homozygous recessive, but it could be it has to be homozygous. So if a plant or animal is showing the same thing for many generations, that would be called as true breed. Got it?
Yeah.
One more terminology is a test cross. Very important, yet very simple. Test cross is a cross done to know the genotype of an organism showing a dominant phenotype. Dominant phenotype. Let's suppose, there's a rabbit, and rabbit is showing 2 colors. The black is dominant, and the white is recessive. Black is dominant and the white is recessive. Okay. Now, what do you think? Let's suppose that our alphabet is B. Dominant alphabet is B and the recessive alphabet is B. Obviously, if a rabbit is showing a white phenotype, what do you suppose its genotype should be? If a rabbit is showing the white phenotype, what do you suppose its genotype should be?
Um, so recessive B, BB, recessive BB.
Homozygous recessive. Homozygous recessive. Complete name is homozygous recessive. But if someone gives you a black rabbit, can you tell its confirmed genotype?
It could be either heterozygous dominant, or
I'm sorry, again?
it could be heterozygous dominant,
Yes, yes, heterozygous and
um, just dominant.
homozygous dominant.
Okay.
Now, you don't know. You only know the phenotype. How would you confirm that it's either a heterozygous or a homozygous dominant? That kind of cross is called as test cross. Read the definition again. A cross done to know the genotype of an organism showing a dominant phenotype. If someone gives you a black rabbit, you cannot say for sure that what is the genotype of this rabbit. Obviously, this rabbit can have a heterozygous and a homozygous dominant. So what you should do? Cross the organism showing dominant phenotype with the one showing recessive phenotype. What would happen? Let's suppose, let's say, let's say the black rabbit, the black rabbit is showing homozygous dominant. Obviously, you are not sure. You need to make sure. So, capital B cross small b because the recessive phenotype is white. So small b or lowercase b or homozygous recessive. So what would happen to the gametes? Let's form the gametes. Remember that if the gametes are of same types, only make one of them. So B is one gamete from this parent and small b is gamete from this parent. Now their F1 would show 100% black rabbits. This would be a proof. This would be a proof that the black rabbit is showing, is having homozygous dominant genotype. Did you understand what happened there?
Um, Not
Okay,
exactly.
okay. Let me draw the next cross and then you'll understand it better. Let's say the black rabbit, let's say the black rabbit had a heterozygous genotype. Now, what is our rule? To know the genotype, cross the organism showing dominant phenotype with the one showing the recessive phenotype. So let's suppose the black rabbit is heterozygous. we are going to cross it with the recessive one. So homozygous recessive. Let's make the gametes, because now now the alleles are different, so we'll show two gametes here, but over here the alleles are same, so we'll show one gamete. Now if this sperm fertilizes this egg, it would be a black, and if this sperm fertilizes with this egg, it would be a white. It means how many chances are there? The chances are 50% white and 50% black. Okay, it means that in a test cross, if you are provided with an organism showing the dominant genotype, dominant phenotype, and you don't know the genotype, just cross it with the one showing the recessive phenotype. If their offsprings are 100% dominant, it means that the black one is having both alleles dominant. We are basically trying to prove this genotype. If the offsprings are 100% black, it means that the, the unknown genotype is homozygous dominant. And if the offsprings are showing 50% black and 50% white, it means the unknown genotype is heterozygous. Is that better now?
Yes.
So that is what the test cross. And we do it a lot. Let's suppose we do it with the flowers. There is a flower having purple color as dominant and white color as recessive. White color as recessive. So what alphabet should we choose? We should be choosing the alphabet that that shows the clear difference. So let's say R is the dominant one and small r is the recessive one. So if you have a purple flower in your hand, how would you identify that what is the genotype? Obviously, it could be it could be homozygous dominant or it could be heterozygous. So you would have to cross it with the homozygous recessive to see the ratio of the offspring, because ratio of the offspring would prove the genotype. Got it?
Yes.
Okay. Punnett square, pedigree damage. All right. Okay, next we are going to talk about the okay. See, the final ratios, the final ratios of genetic crosses uh, depend on the parents. Let me change my sentence. The final ratios of genetic crosses or offsprings depend on the parents. Depend on the parents. Some crosses show, if you remember a previous cross, we did the cross between the two heterozygous organisms. And in this, these are the two gametes, two types of gamete they would be forming. We'll draw a Punnett square and will be showing their probability. In this case, the phenotypic ratio is constant. What is this? In this case, the phenotypic ratio would always appear to be 3:1. But To bring 3:1, the parents must be heterozygous. Must be heterozygous. But if one parent is heterozygous, another one is homozygous recessive, then this parent would be making two types of gametes and this one would be making one type of gamete. Now the ratio would change. And now the ratio is 1 1. So read the sentence in the beginning again. I said final ratios of genetic process depend on the parents. So if there is a heterozygous genotype, 100% there would be a ratio of 3:1 or 75% to 25% result. But if one of the parent is heterozygous, the other one is homozygous recessive, there would be the ratio of 1:1. Is that clear?
Yes.
Okay, now basically the ratios can change. The ratios change. And you know, the three-word phrase or the three-word sentence that I have written over here, this thing have bothered the scientists for quite a long time. Quite a long time. Why? See that when the genetical research was started, uh, Gregor John Mendel, he was a genius scientist. When he started working, he saw that he proved many points and he proved many ratios. Why am I saying that heterozygous ratio would always come out to be 3:1? It's because of the Gregor John Mendel. It is said by the Gregor John Mendel that the cross between the two heterozygous parents would always come out to be 3:1. Or maybe the cross between one homozygous recessive and one heterozygous would always be 1:1. And he showed many other crosses. When he started doing the crosses above monohybrid, he showed many other crosses, many other ratios. Because we are only studying the monohybrid ones in which we only study one character. When we study more than one, the ratios obviously would keep on changing. So when we talk about the ratio changing, there is another thing that hits the concept of ratios, and that is linkage. But for linkage, you need to understand two subtopics. Two subtopics have to be very much cleared. Okay. Mm, One topic is somewhere around here. State that sequence of basis. Yes. Inheritance of sex in humans with reference to X and Y chromosomes. So basically, this is one of the subtopic is this one, and the other subtopic that would help us understanding the changing in ratio is this one. Okay, let's refresh our concept of chromosomes. See, chromosomes, they are made by DNA folding. We fold the DNA, we build the chromosomes, and the genes are attached to the chromosomes. The proper word here is that genes are linked to chromosomes. One of the very obvious thing about the linkage is a gene present on one chromosome can never be found on any other. A gene present on one chromosome can never be found on any other. That is called as linkage. That is basically called as linkage. It means that you know very well that we have 23 plus 23, and you know that the chromosomes from one set of 23 are exact copies of the other set of 23. So if a gene is associated with chromosome number 1, you cannot find it on chromosome number 2, 3, 4, 5, 6, 7, 8, 9, 12, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. You can never find it. Yes, you can find it on its homologous pair. Why? Because we have established the definition of homologous pair, that these two chromosomes are exact identical to each other in their size, structure, and genetic arrangement. So due to being Homo sapiens, we should know that our genes can only be found on one gene can only be found on like homologous chromosomes. If I have one set, homologous chromosome can only be made when you have two sets. You know that. When you have two sets of 23 and 23, you would be grouping them in pairs to make the homologous pairs. But if a gamete, obviously a gamete possesses haploid set. A haploid set is possessing only one set. Now, if you find a gene on one chromosome, you would never find it on another chromosome. That is called as linkage. Did you understand it clearly?
Yes.
And did you understand why I said that that you can find a gene on its homologous pair?
Yes.
Yes. So now the thing is, we have two types of chromosomes. Obviously, we have 23 pairs, but we divide our chromosomes into two groups. Uh, 22 pairs and the last pair, or 23rd pair. The first 22 pairs are the first 22 pairs are are called as autosomal chromosomes. They are called as autosomal chromosomes, and they typically you can say define our body characters. Our body characters. They are called as autosomal chromosomes. The 23rd pair is called as sex pair. It basically the sex of human, defines the sex of human. And in the 23rd pair, it is the only task. It is the only task. This phenomena is called as sex determination by chromosomes. Sex determination by last pair of chromosomes. The last pair of chromosomes often define our, you can say, it often defines our sex. Why? Now you have to be very careful. All autosomal under microscope, under microscope, all autosomals look like this. You know, we resemble them with the letter X. So if there is chromosome number 1 and 1, we call them XX. 2, 2, we call them XX. 3, 3, we call them XX. About the sex pair, about the sex pair in males, one chromosome, meaning 45th chromosome, looks like this, but 46th chromosome looks a bit like this. Upper arms are very short. So under microscope, we call them X and Y. But in females, the last pair or the sex pair is as same as the autosomal chromosomes, meaning both of the last pair individual chromosomes would be appearing like XX. So it means if I say that how do chromosomes, if I ask a question that how do chromosomes determine our sex? So if the last pair combination of chromosome is XX, then it's a female. And if last pair combination of chromosome is XY, then it's a male. You got it?
Yes. Can you also give me 2 minutes? I just need to use the washroom.
Sure, sure, sure.
Yeah.
Okay. So I'll revise the last thing I said, that if the last two chromosomes appear like XX, they would be it would be a female. But if the last two chromosomes appear like X and Y, it would be a male. Now, what are the chances? What are the chances that the next child is a male or a female? Okay, if you are crossing two parents, male cross female, if you are crossing two parents, uh, obviously right now we are not talking about, remember, we are not talking about alleles right now. We are talking about chromosomes. The male chromosome, the last pair in male is XY, and last pair in female is XX. And you know that every pair has to be separated in every pair has to be separated in meiosis. Separated in meiosis, which clearly tells us that X would be separated from Y, and this X would be separated from the other X. I'm only writing it once because they are same. So if the X containing sperm fertilizes the X egg, it would be a daughter. If Y containing sperm fertilizes the X egg, it would be a son. So what chance do you have? 50%. Let's suppose, let's suppose I say that there is a 50% chance of a child to have attached earlobes. Attached earlobes. What are the chances? Carefully listen to me. What are the chances that the child would have attached earlobes, attached earlobes, and a son? Can you tell me?
Um, 25%
Why did you divide the chance of attached earlobe into half?
because of the gender.
Yes, you did
50%
right.
is
You did
male,
it right.
female, so 25 for
Hmm.
boy.
It means the other share has been given to the females. Like 25%, sorry, yes, 25% chances of a son with attached earlobe, 25% chances of a son with unattached earlobe, 25% of a chance with of a girl with attached earlobe, and 25% of the chance of a girl with detached earlobes. So yes, you were right. Got it?
Yeah.
Now, getting back to the previous concept, linkage. The genes, the genes linked to autosomal chromosomes. Chromosomes. Autosomal mean first 22 pairs. Autosomal chromosomes are called as autosomally linked genes. Autosomally linked genes. A note has to be mentioned here that these show same behavior. These show same behavior of inheritance in both males and females. Both males and females. Why am I writing this sentence over here? Because the genes linked to either of the sex chromosome are called as sex linked, meaning that you would never find them anywhere else. Sex linked, and they show different inheritance pattern in males and females. In males and females, which would change our ratio expectations. See, this is the thing we started our discussion from. The autosomal genes won't change the ratio, won't change final ratio. No, they won't change because their ratio is expected, as well as they are going to show the same pattern, same behavior of inheritance. But the chromosomes which are sex linked, they would be showing different inheritance behavior um, in generation, while getting transferred from one generation to the next, and they would deviate from ratio expectation. Okay, first of all, way to write way to write the genes. How do we write autosomally linked genes? Autosomally linked genes. We mostly write them like AA, BB, DD, like this, isn't it? Or maybe we also can write them IA or IB to show multiple alleles, isn't it?
Yes.
But these are autosomally linked. What about the sex linked genes? To show the sex linked genes, first rule, draw the alphabet of chromosome. Draw the alphabet of chromosome like X, like X. First step. Second step, write the gene as the power of chromosome, of chromosome. Meaning, let's say the gene is B. So I would write XB. It resembles a lot with the multiple alleles. But I would write a note here. Don't confuse with multiple alleles. With multiple alleles. Because the main alphabet could only be X or Y in sex linked characters. If the alphabet is X or Y, it is obvious that it's a sex linked character. Is everything clear here?
Yes.
Now, how does it change our expectation of ratios? See, now we are going to talk about a character called as red green color blindness. Red green color blindness. This the gene for basically we have three cones, if you remember. We have three cones: red, blue, green. The gene for blue is present on autosome. It's an autosomal character. But the gene for red cone and the gene for green cone are present on sex, they are sex linked, and in the sex chromosome they are only located at X. Only at X. Remember, another very important terminology here. Those that you find on X, you would never find on Y. Find on X would never be found on Y. Found on Y. Okay. So remember that red and green genes are sex linked. What does it mean? It means that the blue cones show equal behavior in males and females. Like you can expect that the blue cones can show the ratios of 3 is to 1 or 1 is to 1. But let's see. Uh, the dominant allele or a normal person, a normal person would show a presence of an allele like uh, capital R. It means he got no color blindness. How would you write it? XR. A person with the recessive allele, you would say diseased person, would be possessing a small r, meaning that this person is colorblind, and you would write it XR. Let's say uh, we cross a male and a female in which the genotype of a male is XRY and the genotype of a female is XRX small r. Why am I not writing a gene on Y?
I'm not sure.
I said, okay, I said that if you find a gene on X, then it is never going to be present on Y. It means that it is present on sex chromosome, but in sex chromosome it is present on X, not on Y. So you would write it XRY. Why? We won't write the gene on Y because the R is only X linked. Obviously, it is sex linked, but in sex linkage, it is present only on X, not on Y. You got this point?
Yes.
All right. So now X capital R Y, X capital R, X small r. Now let's draw the crosses. X capital R, X small r into X capital R Y. Okay, so X capital R and X small r both would be going into different eggs. X capital R going into different egg, Y going into different egg. If we draw the Punnett square, X R, X small r, X capital R, Y. Now this is what, a daughter, right? And this is homozygous dominant, like it, she has no chance of having color blindness. Next, a son. Would this person be having a chance of color blindness?
Um,
Look what I have written in the front of capital R.
so no color blindness would be capital R.
Yes.
So there would be a 50% chance here.
Let me tell you. Now, let's talk about X small r fertilizes the sperm with the egg with the fertilizes with the sperm with the X capital R. So is there a chance of color blindness? Still no. Why? Because there is a dominant one present. But now X
Now there
small
is a
r y.
chance. Yeah.
What is the difference here? You know, phenotypically there is no difference. The ratio is same, 3:1. Don't you think?
Yeah.
But the pattern that is changing is basically the appearance of color blindness in males and females. Previously, in any character, we did not say that this character would be different in males and this one would be different in females. But now we are saying that this character would show different behavior in males and different behavior in females. That is why the ratios deviate, because the genes are sex linked. And remember that we must know that if the genes are sex linked, they could be present only on X and only on Y. If they are present on Y, you would never find them on X. And if they are X linked in sex linkage, you would not find them on Y. Now, let me write a sentence over here. For X-linked diseases, for X-linked diseases, males are more vulnerable. Males are more vulnerable than females. What do you think the reason of it? Going to the previous sheet, try answering this question. Males are more vulnerable than females. Can you tell me?
Because the male one has only a dominant allele, and the female has a balance of a dominant and recessive allele.
You can say that females can have the alternate of a recessive allele, while the male doesn't have any choice related to the alternate of an allele. So that is why these diseases appear more in males than females. Females are mostly prevented because they actually are, like, you can simply say, they actually are really they have an alternate. They can easily just look at bring an alternate allele and cover up the effect of a recessive allele, but males don't have that chance. Got it?
Yeah.
All right. One small topic of this chapter, and then we are finished with it, and that would be this 12th one. So not no sorry, not this one. It would be this one. This requires a bit of attention, but it is not that much of a difficult. I'm about to send you another worksheet. Okay, I'll be expecting both by tomorrow. Okay.
Okay.
All right then.